Good morning, everyone. And it's the third day. I think people are getting tired. Anyway, so to start, keep the program. I'll ask Fernando Tuya to give the third day's plenary lecture. I'll leave the floor to him. So we have 30 minutes.
Okay, good morning, everybody. First of all, I would like to thank all the organizers for giving me the chance to be with all of you today. So basically here in my presentation of today, what I'm going to do is I'm going to present a summary of a compilation of the results that our research group have been doing in the last couple of years. And at the same time, I'm going to present those research lines that we are tackling at the moment. The idea is to provide an holistic overview on how we should tackle the study of an entire ecosystem. In this particular case, the study of seagrass meadows dominated by the seagrass Cimodocea nodosa. So I think that we all here in this room, we all love the nature, and we as humans need to define certain biological entities to facilitate the study. And this is why we have defined ecosystems as entities that encompass a set of organisms living within a particular spatial context, and at the same time, the physical medium where these organisms live. Two key elements of any ecosystem that we normally tackle for any study is the functioning and the resilience. In other words, how a particular ecosystem functions, and which is the resilience of this ecosystem under different types of perturbations. And we have developed a whole range of different attributes and descriptors to characterize either the functioning or the resilience of the ecosystem. For example, for the functioning, we have defined we can consider primary productivity, secondary productivity, nutrient recycling, and different attributes as the biomass of whatever, density, diversity, etc. In terms of resilience, we can measure the resistance and recovery for varying perturbations of different intensity and frequency. And of course, as I highlight here, we can always be totally overwhelmed by the massive amount of descriptors and attributes that we can measure. Okay, so let's take a case study example. This beautiful forest dominated by Cimodocea nodosa, which, as I say here, can be considered as a foundation species in the sense that it provides a three-dimensional habitat for a range of organisms that find shelter and food within these canopies. One of the first things that we can do to study any ecosystem that is underpinned by a foundation species, as this seagrass, are the temporal tendencies, how this ecosystem has been fluctuating through time. We can do that through the measure of different attributes. For example, here for this particular seagrass, we have the density of shoots, we have the leaf length, and we have the cover between different years. For example, here between 2003 and 2012. And we also can use maps that basically give us an idea of how habitats have been changing and shifting through time. Okay. All of this should be done, for example, in the framework of different monitoring programs, okay? So that's why it's quite important, as Steve was saying yesterday, that we have solid monitoring programs, okay, to get this information. We also can do another type of approach. We can do somehow like meta-analysis. We can compile every single data, for example, relative to any foundation species, for example, in this case for Cimodocea, and we can work out trends through time for the different descriptors of the plant. For example, here you have how seagrass density and how seagrass cover has changed for all the island during the last 15 years. And so some patterns emerge, some patterns pop up. Here, for example, it's clear that shoot density, for example, at the most populated island, as Tenerife, Gran Canaria, Fuerteventura and Lanzarote tend to decrease through time, while on the other hand, in the less populated island as El Hierro and La Gomera, there is basically no trend. Okay, it's very important to realize that when we tackle any study, we can approach the problem according to the different levels of organization. So, for example, in the case of our case study, we know that cells, they form tissues, and tissues form the basic functional unit of seagrass meadows, which are the seagrass shoots. A collection of shoots, of ramets, are going to form a seagrass patch, and a set of different seagrass patches are going to create meadows that normally are encompassed within large landscape that may be connected with other type of habitats, as for example, rocky reefs, coral reefs, sandy bottoms, whatever. So if we want to tackle any issue from a holistic point of view, we always should be remind this hierarchical organization of nature, right? So, for example, at the plant level, we can study its functioning according to either physiological, morphological, and demographic attributes. One of the problems that we encounter when we study any clonal plant, any clonal organism as this seagrass, is that we don't know where the organism is starting and ending. Okay, as I said yesterday, seagrasses are clonal organisms, so we don't know, contrary to, for example, a palm tree where we have one individual and another individual, we don't know where an individual starts and where it ends. So that's why we always need to incorporate measures of the genetic identity of the organism, right? And this is why we need to include genes as a driver of the responses of in this particular case of the plant, when we study either the functioning or the resilience of the foundation species, in this case of the seagrass. As I said before, seagrasses are clonal plants, so we know that in theory we can expect a connection between how the genotypes distribute in the bottom and how the patches are distributed. Okay, in theory, and that's why we need to incorporate ecology with genetics, and that's why landscape genetics needs to meet landscape ecology. Okay. And this is basically going to bring along a couple of really important questions in terms of the management, conservation of this ecosystem. For example, we can wonder if different clones physiologically work differentially, if different clones demographically differ, and importantly, and this is a quick question in terms of conservation biology, if the resilience of the different clones is different, okay? And at the end of the day, all of these questions are going basically to translate in one really relevant question, and is if different genotypes translate into different phenotypes, okay? Okay, so as I said yesterday, at this point, taking into account all the things that have been pointed out, I have to self-criticize my experiments in the past, because when I try to work out which are the effect of a range of environmental stressors, for example, fertilization, sedimentation, blah, blah, on my case study species, in this case, the seagrass, I always have ignored the genetic identity. In other words, I've been setting up my experimental plots, and I don't have a clue if I'm working with one genotype, with one individual, I'm working with two individuals, I'm working with three, and so on. So I basically have been treating the genetic identity as a black box, okay? So that's why I think that we people working with clonal organisms, we always need to incorporate genetic information to work out responses, okay? At the end of the day, it's a way to decrease the amount of noise, okay? It's also important to keep in mind that if we have, for example, 3 seagrass meadows, as I have here, they can be constituted by a certain amount of genotypes. Okay, imagine, for example, that this is constituted by 2 genotypes, this by another by 3 genotypes, and this one by another 3 genotypes or whatever. But these seagrass meadows, they can differ in terms of the number of genotypes, but they also can differ in terms of the genetic relateness among them. Okay, so it's not only a matter of the number of genotypes, the number of clones that constitute a particular seagrass, but it's also a matter of the genetic relateness among them. And we should tackle this problem by a range of genetic measurements, as for example, the expected or the observed heterozygosity. Okay? So, at the end of the day, what we want to do, and this is something that we are doing at the moment, is trying to correlate whether the functioning and the resilience of seagrass meadows are connected with different measurements of diversity. Okay? And basically, by addressing this issue, we are going to be able to address one key issue in seagrass conservation at the moment, and is if an increased genetic diversity is going to increase resilience against a range of disturbances, right? So, as I said yesterday in the workshop, we can tackle this issue by different approaches. We can, of course, set up experiments in the field working with seagrass meadows that we already know that have different types of genetic diversity, but we also can do manipulation in mesocosm, and we can work with monoculture and polycultures, okay? And all of these things that I've been doing, they all can be complicated in the sense that any species, as this foundation species, have a particular distribution range. So, for example, our case study species Cimodosia is distributed across the entire Mediterranean and from southern Iberian Peninsula to the west coast of Africa. So, all of these genetic patterns are going to be modulated by geographical and evolutionary determinants, and it's key, therefore, to incorporate not only local variations in genetic diversity, but also variation across the entire distribution ranges, okay? So, at the end of the day, from a methodological point of view, this means that we have to set up experiments at certain locations and replicate these experiments across the entire distribution range, okay? So this way we can disentangle local and regional scale determinants. And in turn, this way we can also reply another quick question in conservation biology. If there are differences in the functioning and in the resilience of foundation species between edge and central population, okay? And this is something that we are doing at the moment, that with this project I was presenting yesterday. So we basically are going to set up the same experiments at a range of locations across the entire distribution range. Okay, I've been speaking a lot about prospective lime, which somehow are tricky. So now I'm going to move to more common things that we have been doing in the past. We know that the plant is not only a plant, in the sense that the plant is providing shelter and food for a wide range of organisms. For example, if any of us go to any seagrass meadow, we know that we are gonna find a larger diversity and a larger abundances of creatures relative to unvegetated bottoms that are just a few meters away from the seagrass meadows. For example, our results for here for the Cimodosia nodosa seagrass meadows has been indicating that we can get densities of invertebrates larger than 1,000 invertebrates per square meter. And we know that these large densities, they can fluctuate through time. For example, following the natural life cycle of the seagrass. At the same time, the seagrasses are not only providing food and shelter for invertebrates, they are also providing food and shelter for key assemblages as fish. For example, in our seagrass meadows, we can reach up to more than 60 species of fish species. 40 of them have commercial interest. One of the key functions that we have described for these seagrass meadows in relation to fish is the key fish nursery function. As a data, I can tell you that about 95% of the fish that we find in a seagrass meadows are recruits, are juveniles of different species. Okay, so we find a lot of fish in seagrass meadows, but more of the fish are small sized fish. Okay, in other words, seagrass meadows are the place where a whole range of species develop their early life cycles. Okay, and this is what I like to represent with this scheme here, okay, where we have a tiny fish sleeping in the middle of the canopy. The fish nursery function is only one relevant function, and it's very important that we keep in mind all of these functions, because at the end of the day, these functions are underpin ecosystem services. Okay, so for example, in the case of seagrass meadows, we can define a whole range of functions. We know that seagrasses, they can sequester carbon, they can export organic matter to adjacent unvegetated habitats. They are also going to contribute to nutrient recycling. They are going to produce oxygen. They are going to contribute to sediment stabilization. They are going to dissipate waves. And at the end of the day, this is going to provide a key ecosystem services, which is an improved water quality. And we also know, as I said before, that seagrass meadows are going to provide food and shelter for a range of organisms, and they are also going to contribute to the recruitment of a whole range of animals. So, as I say before, it's key that we always try to work out the different functions of any ecosystem, because they are going to underpin the ecosystem services. Okay. In the particular case of seagrass meadow, we can divide the different ecosystem services in 4 big groups. We have, first of all, supply services. We have regulation services. Third, we've got these habitat services. And finally, we have cultural services. So one of the trends that in conservation biology has been developed in the last couple of years is trying to work out which is the financial value of these services. Okay, and this can be a little bit tricky sometimes. I mean, to give nature a value, an economic value, but from the point of view of managers, it's quite valuable. Okay, and it's not only valuable for managers. I personally believe that it's also important for society, because it's a way, it's a language that they are going to understand, which is the value of the ecosystem. I'm going to give you some examples of some studies that we have done in the past. Let's take as an example the parrotfish, Parisoma cretense, which, as you may know, is an icon of our Canary Island cuisine. By taking advantages of our monitoring program, where we can estimate the abundances and the biomasses of the parrotfish, by using the size frequency distribution and by using the allometric relationship, there are a collection of formulas and algorithms that are going to provide measures of production per area and per unit of time. If we take into consideration the amount of seagrass meadows that we have in one island, for example, in Gran Canaria Island, we are able to estimate the value of that particular production by taking into account the value, the market prices of these target species. In the particular case of the parrotfish, we have estimated that the value of this production per year, taking into account the entire island perimeter, reach €55,000. Okay, and this is just for one species, 1 year. Okay, imagine that we keep on going doing this for the whole range of fish species that we find in a seagrass meadow. Okay, the value is going to increase dramatically, right? And keep in mind again that this is only taking into account the value of seagrass meadow as a fish nursery place. Okay. Why I'm saying this? Well, I'm saying this because we know that in any seagrass meadow, we can find a whole range of fish, a whole range of organisms. So again, if we take advantage of our abundance and biomass estimators, if we take into account the area that is covered by the seagrasses in a particular area, we can, for example, estimate which is the commercial value of that entire biomass. To give you some examples, we have estimated that the value of 1 hectare of seagrass meadow as a fishery value reach up to 1,700 euro. Okay? So if we take into account the entire area that is covered by Cimodosia seagrass meadow at one particular island, in this case Gran Canaria, the value of the seagrass meadows can reach more than €1 per year. Okay? If we assume that half of this biomass is going to be collected, the value of this biomass would be a little bit more than €500,000. Right? Okay, so I've been talking about ecosystem functions, ecosystem services, ways that we can estimate the value of these ecosystem services, and now some of the other things that I would like to communicate, to get across is that is the idea that some of these ecosystem functions and services can be eroded, can be decreased. For example, we know that when there is a deterioration in the condition of the environment, these seagrass meadows, they can shift towards an alternative state which is dominated by seaweed. Okay, so for example, when there is a deterioration associated with an increase in nutrients, increases sedimentation, increases turbidity, we know that the ideal ecosystem state, which is dominated by seagrasses, shift towards a state that is dominated by seaweeds. Getting back to our case study, we know that in the Canary Island there has been transitions from bottoms that were initially dominated by Cimodosia to bottoms that are actually dominated by a green algae, the green algae Caulerpa prolifera. And so one of the questions that was raised some time ago was, okay, if there is a replacement of Cimodosia by Caulerpa, is there a functional redundancy in the provision of these functions and these services? In other words, what is going to happen, for example, with the primary productivity, with the secondary productivity, with the provision of habitat for small fish, and all of these kind of things? So we tackled this issue some time ago, and we finally managed to quantify the erosion in this ecosystem function when we compare a meadow dominated by Cimodosia relative to a meadow dominated by Caulerpa. So here I'm providing some of this data. For example, we know that primary production, net primary production, is eroded 1.5 times. We know that the abundance and diversity of epifauna decreased between 1.7 and 3.1 times. We also know that the abundance and diversity of small-sized fish decreased between 1.8 and 2.6 times. The abundance and diversity of large-sized fish decreased from 1.6 to 3.6 times. And we also know that sediment stabilization is decreased by 1.4 times. So what is all this information telling us? That there is an erosion in ecosystem functions when we compare a meadow dominated by Cimodosia with a meadow dominated by Caulerpa. In other words, if we have a Cimodosia nodosia seagrass meadow and is replaced by a Caulerpa meadow, there is going to be an erosion in the functions and so in the services that provide us, right? So, as the take-home messages of my presentation, I would like to stress 2 points. First of all, we always need to include measures of genetic diversity when we work with clonal organisms, in particular when we want to describe and to work out the responses versus a whole range of disturbances. And second, that we need to recognize ecosystem functions to aim to valorize, at least financially, or maybe economically, these ecosystem services. That's all. Thank you very much for your attention. Yep.
Thank you. Thank you for your talk, Fernando. Question: when you in the slide where you had the geography, the biogeography of the seagrass, what do you mean by the center and side population? What is this? Can you define?
Oh, I can use this. Yeah. Well, there is actually a paradigm in ecology and conservation biology that basically says that population tend to increase in abundance and in fitness in the center of the range. There should be a decrease in the way they behave. I mean, in terms of fitness, and in the abundances toward the rear of the edges. I say this is a paradigm basically because there is no proof at all. I mean, it works for some species, it doesn't work for other species. But at least it has been pointed out, as a relevant hypothesis to test whether central and rear populations behave different or not. I think in this hypothesis in terms of management can be really important because if, for example, the resilience of seagrass meadow differ between central and rear population, management measurements can differ. For example, as we were discussing yesterday, we know that for Cimodocea in the Mediterranean, it's very resilient to any type of disturbances in terms of sedimentation, silting, fertilization, and so on, while in the Canaries, the resilience is much decreased. So I wonder, is this a causality that Cimodocea here in the rear of its distribution has less resilience than in the Mediterranean, which is in the center of the distribution? Maybe yes, maybe no. So this is some of the questions that we are trying to tackle in that sense. So that's why, we always try to respond this kind of question because at least from a theoretical point of view, they are important.
How important the density-dependent processes in the
which one?
Density-dependent processes in the demography of these seagrass beds, and do you get differences in density dependence with different clones?
Well, that's a really good question. We actually don't know. I mean, probably there is a compromise between facilitation and competition because seedlings, for example, they can facilitate by adult plants in the sense that they are going to grow in a more calm, tranquil environment. For example, they are going to give protection against waves, and they are also going to give protection against grazes. But at the same time, there is a limitation because of light, because the amount of light that they are going to get is going to be less. It's something that we still don't know. We would like to address this with this project that we got, but at the moment it's undecided and it's unknown.
So do you think there's a minimum density required? Positive density?
Absolutely. I think it's a local thing. I think that in some circumstances, at some sites, at some particular window frames, times, the adult canopies can facilitate the seedlings and can facilitate recruitment. In other cases, it can be the other way around. So I think it's a really local thing. At least that's my first impression. And there's huge variability from one side to the other.
Tuya, when you talk about what Gerson asked, the middle and the edge populations, you mean the whole geographic distribution or one meadow?
I say it again. When you
mentioned the health of the ecosystem or the plant.
Yeah, well, what I was saying is that it's key to study the functioning and the resilience of, in this particular case, seagrass, Nodosia meadows, at both local scale and regional scales. So that's why we need to replicate studies and experiments, not only at local scales, but also across the entire range. So we can somehow disentangle mechanisms that operate at small scales from mechanisms that operate at large scales, because they can be totally different. At local scale, for example, herbivory might be important, while at large scale, the evolutionary context of the evolution of the species can be important. So there are a whole range of mechanisms that operate at different scales, temporally and spatially.
Thank you very much. For any other questions from anyone here? I have one.
You first.
No. Are you trying to get a handle of above ground and below ground processes in terms of ecosystem functioning and potential for services?
That's another really good question. I think at the moment we are mainly tackling, above ground processes, but I totally agree that below ground are as important as above ground processes. For example, it may happen that after a disturbance, all the canopy is removed, but if the below ground stand is going to recover. So that's another key issue that we want to address. In terms, these experiments I was saying before that are going to be replicated in the entire range, want to test this. So we're gonna manipulate different types of mechanical disturbances. In one case, it's just removing the canopy. In some cases, removing the canopy and the below ground material. So we are trying to unravel this.
So
you?
No, it's okay. No. I'll talk to you later because we've got to keep.
Okay.
It's about nutrient cycling and so on. Okay. So thank you very much again, Fernando.